everyone, welcome to TryRight. This is Pack 365, myself Srija. So today in this session, you're going to learn about CPT coding guidelines for pathology and laboratory. So what are the guidelines involved in pathology and laboratory? So what are the sections classified in this criteria? So first is genomic sequencing procedures. So what are genomic sequencing procedures? So in shortcut, they are called as GSPs. So they are the lab tests that analyze DNA and or RNA to look at many genes at the same time. So instead of testing one gene at a time, these methods check multiple genes or even the whole genome in a single test. So old method is reading one page, GSP is scanning an entire book at once. So these tests, they are used when a patient's condition might be caused by genetic changes and we don't know exactly which gene is responsible. So what do this test actually examine? So they can look at the specific gene panels. So what are specific gene panels? They are a selected group of genes linked to a disease. For example, a cancer gene panel may test for 50 to 500 genes known to cause the tumor growth. So these are used when the disease type is known. So there are common genes associated with it. So next, exome sequencing, WES. So looks at all the exons, the protein coding parts of the genes. So exons they are the only 1 to 2% of the genome, but they contain similar to 85% of the known disease causing mutations. So these are used for the developmental delay, intellectual disability and the rare genetic disorders. Genome sequencing, WGS. So the analyzes the entire DNA sequencing. The most comprehensive test can detect the small mutations, structural changes and the variants in the regulatory regions. So now what are the technology used in genome sequencing procedures? MPS, massively parallel sequencing. These are also called as next generation sequencing, NGS. So this is a key technology that makes GSPs possible. So how it works, instead of sequencing the DNA one fragment at a time, MPS sequences millions of fragments simultaneously, like scanning thousands of barcodes at once instead of one by one. So what are the benefits? Faster, they are cheaper than the old sequencing, so they can also analyze the large amounts of genetic data. So now what are the type of samples that are used here? So GSPs, they analyze the nucleic acids like DNA or RNA from the germline samples from normal body cells such as blood, saliva and cheek swab. They are used to find the inherited genetic conditions. And then neoplastic samples. So these are from tumor tissue. So they are used to find the cancer related mutations. So these mutations, they may help choose targeted therapy. They predict the prognosis and they determine the eligibility for the clinical traits. So what are the clinical applications here? Fetal testing. So for fetal testing, the cell-free fetal DNA from maternal blood is taken. They used to detect the Down syndrome, trisomy 21, trisomy 18 and trisomy 13. So this is called as NIPT, non-invasive prenatal testing. And the second is cancer testing. So gene panels detect the somatic mutations in tumors such as EGFR, lung cancer, BRCA1, 2 marker genes for breast or ovarian cancer, and KRAS, BRAF, ALK, etc. So they help in precision medicine. And also coming to the developmental disorders. So whole exome or genome sequencing, they can find the genetic cause of developmental delay, autism spectrum disorder, intellectual disability, and also congenital anomalies. So why exome and genome tests, they are special? So exome and genome procedures, they evaluate the genetic material in totality or near totality. So what does this mean? They mean they are broad, unbiased tests. So they are used when doctors know which gene is causing the problem. So it is helpful for rare or complex conditions. So next subsection, chemistry. So this means most chemistry test codes do not restrict specimen type. So how to report this test correctly? So blood, urine, CSF, body fluid, etc. they can be used unless the CPT code specifically says otherwise. For example, glucose can be measured in blood, CSF or body fluid, but you have to report the same glucose CPT code unless the code specifies a different source. For in the case of multiple specimens where multiple reports will be there, so if the same analyte is tested from different sources or different times, you have to report it separately. So an analyte, what is it? It's a substance which is being measured. For example, like glucose, sodium, potassium, etc. So you have to report the separate CPT codes when the situation is same test and different specimen types. So you have to report the bill each specimen here. Whereas in same test, different times, you have to bill each time. So one example is glucose from blood is taken and also glucose from CSF. So you have to report glucose twice. And example 2, troponin measured at 8 a.m., 2 p.m. and 8 p.m. at different times. You have to report the troponin 3 times here. So this is how you have to report the situations and the time given. So chemistry tests, they are always the quantitative, usually. So the examination is quantitative unless it is specified. So this means most chemistry codes, they measure exact amounts. So quantitative, it gives a number. Whereas qualitative, it is just positive or negative, like a pregnancy strip test. So if a test is qualitative, the CPT description will usually say so. So what are the organ or disease panels? So these are the code ranges 80,048 to 80,076. These are organ or disease panels. So these codes represent the predefined test panels but not the individual analytes. So if all the tests in the panels are done, you have to report the panel code but not the individual test. And then calculated values. So calculated values are not separately reported. So if the lab derives a value from other test, you don't bill extra. So these are considered a part of the main test. For examples of the calculated values are LDL cholesterol. So this is calculated from the total cholesterol, HDL and triglycerides. Anion gap. So this is calculated from the electrolytes. EGFR calculated from the creatinine. So you only bill for the actual measured analytes but not the calculated result. Then next subsection is hematology and coagulation. So this part explains what to do when a test doesn't have a specific CPT code. So when to use the unlisted code 85999. So generally this is used for hematology or coagulation tests that don't fit anywhere else. So you have to use 85999 code when the test is not listed in the 85,002 to 85,810 criteria, that is a hematology section. So if it is not in the chemistry section, it is not in the immunology section, and it is not in the transfusion medicine. So this is called an unlisted procedure code. So what is important about this unlisted codes? So when reporting 85,399 code, you must provide the documentation. You have to include the test name, you have to include the method used, the purpose of the test, the medical necessity of the test, and you have to give the comparison to similar CPT codes. So insurance companies, they review these manually, so the payment is not automatic. And coming to immunology section. So this section includes the tests that study the immune system like antibodies, antigens and immune reactions. So when to use this unlisted immunology codes? 86,849. So if an immunology test, it is not listed in the immunology CPT code range and it is not found in the chemistry range. So then you have to report the CPT 86,849, which is an unlisted immunology procedure. So what are the examples of the immunology test? Autoimmune test, ANA, the rheumatoid factor, allergy testing, IgE antibody, antibody titers and the complementary levels. So if a new or rare immune marker test is performed and there is no CPT code exists, so you have to use 86,849 code. So as with all these unlisted codes, you must submit the documentation which is describing the test performed, the method used, the clinical purpose of the test performed, and similar CPT for comparison. Transfusion medicine. So this area involves blood banking and the compatibility testing. So this transfusion medicine, it includes the blood typing, cross-matching, antibody screening, compatibility testing, and transfusion reactions. So when to use this unlisted transfusion medicine code 86,399. So if the transfusion related test is not listed in 86,850, to 985 code range and it is not in the chemistry or immunology, you have to report this CPT code, unlisted code. So example situations like specialized RBC antigen testing, not in CPT. Advanced compatibility studies, new transfusion related technology test. Again, documentation is required for payment. Coming to microbiology, this section covers the organisms that causes the infection. So bacteriology, the causal organism is bacteria. Mycology, fungi. Parasitology, so the organism is parasite. Virology, viruses. So this slide explains two levels of organism identification, which is very important for coding. The one is presumptive identification. The second is definitive identification. So what is presumptive identification? This is a basic preliminary identification based upon the simple observations. So what are those? It includes identification by colony shape or color, growth on the selective media, gram stains, up to 3 simple biochemical tests. So what are the examples of these 3 tests? Catalase, oxidase, indole, urease. So this gives a probable idea of the organism. For example, the organism is gram-positive cocci, which is a catalase positive, likely staphylococcus. But this is not the full confirmation. When coming to definitive identification, this is the full confirmed identification of the organism to genes level, for example, staphylococcus, and species level, Staphylococcus aureus. So this definitive identification, it requires the additional more complex test such as biochemical panels, automated ID systems, slide cultures such as fungi and molecular methods. So this provides the final reportable identification. So what is the key coding concept here? Presumptive identification, it's a basic and limited testing, whereas definitive identification, it's an advanced testing for precise organism name. So definitive testing justifies higher level CPT codes compared to simple culture observation. So when extra advanced tests are done, so how do you code? What are the additional testing rules here? So if after identifying an organism, the lab performs the advanced techniques, so those are coded separately. So these include the molecular probes, nucleic acid sequencing, chromatography, and immunologic methods. So these are reported using CPT code 87140 to 158 and they are built in the addition to the basic culture or identification code. For example, a bacteria is grown in the culture and then PCR is used to confirm the strain. So you have to report the culture or identification code. And also 87140 to 158 code ranges for molecular probe or the additional method. So what is important here? Do not mix with molecular diagnostic codes. So why? Because molecular pathology section codes are for human genetic testing, not for identifying the microorganisms. So modify 59, which we assign for distinct specimens or distinct sites. So if microbiology testing is done on different specimens or body sites, you have to use modify 59. For example, own culture from leg and own culture from arm. So same test but different sites. You have to append modify 59 to the second code. So what does mean? This is a separate and distinct service. Whereas coming to modify 91, it is assigned when there is a repeat procedure on the same day. So if the same test is repeated on the same day to monitor the patient status, you have to use modify 91. For example, blood culture drawn in the morning and repeated in the evening because infection is suspected. So the second test will always appended with the modifier 91. And there is an unlisted microbiology code 87999. So if a microbiology test is not listed in 87003 to 87912 and not in chemistry or immunology, you have to use a CPT code 87999 code, which is an unlisted microbiology procedure. So generally, this is used for the new organism detection methods and also the rare identification techniques, experimental microbiology test. So documentation, it is required for reimbursement. Anatomic pathology, which includes post-mortem examination. So this section, it shifts from lab testing to pathologist physician services. So CPT codes 88,000 to 88,099, these are the physician services codes. So these codes represent the professional services provided by a pathologist but not the routine lab testing. So generally these codes include the autopsy examination, the gross eye microscopic examination of the organs, determining the cause of the death. So here you have to use the modifier 90. Why? Because if the post-mortem exam or pathology service is performed by an outside lab, you have to use modifier 90. For example, the service was referred to another lab. And there is an unlisted necropsy code. So unlisted necropsy is also called as autopsy. The code is 88,099. So if a post-mortem service is not described in the post-mortem codes, so you have to use when special autopsy techniques are used. So you have to use when there is an unusual post-mortem procedures performed. So again, for this reporting, this unlisted code, you have to maintain the documentation. So documentation is very much required here. And cytopathology. So what is cytopathology? It's a study of individual cells but not whole tissue sections. So this is generally used to detect the cancer, precancerous changes, infections, inflammatory conditions. So what are the common examples included here? It might be pap smear, fine needle aspiration, FNA cytology, and body fluid cytology, pleural and peritoneal. So what is the unlisted cytopathology code here? 88,199. So if cytopathology service is not described in the 88,104 to 88,189. So you have to report the CPT code 88,199 when it is used for new cytology techniques. For also special staining or evaluation not otherwise listed. So documentation must explain the service what has happened. So this is how you have to code the procedures included in the cytopathology. So cytogenetic studies. So what is cytogenetics? It's a study of chromosomes to find the large genetic changes. So generally, this test detects the extra or missing chromosomes. For example, like the condition Down syndrome, translocations, which is common in leukemia, and also the structural chromosome abnormalities. So examples like karyotyping, FISH, and chromosome analysis in the cancer. And unlisted cytogenetic study code 88,299. So the study is not listed in 88,230 to 291 and it is not covered under surgical pathology codes. So then you have to assign this unlisted code. So molecular pathology coding. So this test they analyze the DNA or RNA changes at the gene level. So CPT organizes them into tiers, tier 1 and tier 2. So tier 1 codes they include 81,161, 81,200 to 81,383. This is for commonly performed well-defined gene test. So examples like BRCA1, 2 and CFTR for cystic fibrosis and BRAF gene for mutation. Whereas tier 2 codes, the code range is 81,400 to 408. So these are for less common or more complex gene tests. So they are generally grouped by level of complexity instead of the gene name. And genomic sequencing procedures. So the code ranges are 81,410 to 471. So they are used for multi-gene panels, exome sequencing and genome sequencing. So this look at many genes at once. Multi-analyte assays with algorithmic analysis, which is simply called as MAAA. So the code ranges are 81,500 to 512. So generally this analysis they combine multiple lab tests, computer algorithms, provide risk score or interpretation. So examples like prenatal risk assessment test. So if no code exists, you have to use unlisted molecular pathology codes 81,479 or 81,599. So generally they require full documentation. So these are included under cytogenetic studies. So next is surgical pathology. So this is a study of tissue removed from the body during surgery or biopsy. So examples include biopsy of the breast lump, colon polyp removal and tumor excision. So these codes include, the code ranges are 88,300 to 309. So generally they include a specimen receipt, that is accessioning, gross exam, microscopic exam and written report. So these codes, they are based upon the complexity of the specimen but not the size. So the code range 88,300, it involves very simple specimen complexity, whereas 302 for low complexity, 304 for moderate complexity, 305 for standard biopsy, 307 for major resection complexity, and 309, it is for very complex tumor resection complexity. And the additional services 88,311 to 387 and 88,399. So these are add-on or special procedure codes such as special stains, immunohistochemistry, electron microscopy, and special techniques. So these are reported in addition to the main surgical pathology code when performed. So what is the unit of service? The specimen. So for CPT codes 88,300 to 88,309, you bill per specimen, not per container, block or slide. So what is a specimen here? It's a tissue that is submitted separately, which requires its own examination, which receives its own pathological diagnosis, and one patient can have multiple specimens and each can be coded separately. So example, like a patient has skin lesion from the right arm, skin lesion from the left arm. So they are separately identified specimens. So you have to assign the codes for 2. So 2 or more such specimens are each assigned an individual code. So if the surgeon labels tissues separately, the pathologist treats them separately. Example, 3 GI biopsies from different locations, 2 breast lumps submitted in separate containers. So each specimen must give a separate CPT code based upon the complexity. Now CPT code 88,300. This is for only gross examination. So generally this code is assigned when the pathologist can make a diagnosis without a microscope. Example like a medical devices, pacemakers, screws, foreign bodies. And CPT code 88,302 for gross examination plus microscopic examination, but there is no disease expected. That means it involves the absence of the disease. So this is done mainly to confirm the identity or absence of the disease. For example, hernia, sac. So these are usually benign and routine specimens. And CPT code 88,304 to 309. So these codes are used when gross and microscopic exam is required and the disease is possible or expected. So generally 88,305 is the most commonly used pathology code. So how code levels are chosen? The CPT code book assigns a specific specimens to each level. So you do not choose based upon the size of tissue, number of slides and the time spent. So you have to choose based upon the type of specimen, the expected complexity of the diagnosis. So now what happens if specimen is not listed? So if a specimen isn't specifically listed, assign it to the code that most closely matches the physician work and complexity. So this requires comparing to the similar listed specimens. So what is the unlisted surgical pathology code? 88,399. So generally this code is used when a surgical pathology service is performed but no specific CPT code exists in 88,300 to 387. So unlisted codes do not have assigned RVUs, they require documentation. So they often need comparison to a similar CPT code for payment. So unlisted miscellaneous pathology test 89,240. So this applies to the lab or pathology tests that are not found anywhere else in chemistry, hematology, coagulation, immunology, transfusion medicine, microbiology and cytopathology. So then you have to report 89,240 code. So rare or new tests that do not yet have a CPT code. So why it is different from 88,399? Because it is used for unlisted surgical pathology and 89,240 is used for unlisted laboratory or pathology test. So next is reproductive medicine procedures. So if a reproductive lab procedure is performed and it is not listed in 89,250 to 356 category, you have to report 89,398 unlisted reproductive medicine laboratory procedure. So what are the examples of reproductive medicine procedures? Sperm preparation, sperm analysis and embryo handling. So if a brand new fertility lab technique is performed and there is no CPT code exists, so in these cases you have to assign 89,398 code. And proprietary laboratory analysis PLA. So this is a special category of CPT codes for company-specific laboratory tests. So what are PLA codes? What are PLA tests? So these codes that describe the proprietary lab tests which are developed by a specific company or lab. So they may be performed by one sole source lab or licensed to multiple labs, often FDA cleared or approved. So what are the types of tests in PLA section? ADLTS and CDLTS. So ADLTS, it's Advanced Diagnostic Laboratory Test, whereas CDLTS, it's a Clinical Diagnostic Laboratory Test. So these were recognized under the PAMA Act 2014 to better track the modern molecular and genetic test. So if a PLA code exists, you must use it instead of 81,479 and 89,240. So PLA codes that describe specific brand name or proprietary lab test. So what types of test falls under PLA? MA. So it's a multi-analyte assays with algorithm analysis. So generally, this test measure the multiple biomarkers. They use as a computer algorithm to generate a final risk score or result. Example, a cancer recurrence risk score based upon the expression of many genes. So GSP, genomic sequencing procedures. So this analyzes the DNA sequencing, large panels of the genes, as well as sometimes whole exomes or genomes. And naming convention. So PLA code names try to match existing CPT language when possible. So you might see familiar words like algorithmic analysis, genomic sequencing, and tumor profiling. So this helps you to recognize what the test does. So PLA codes, they are not category CPT codes. So even though they are in the CPT book, PLA codes do not have to meet the category 1 requirements. So instead they have a special inclusion rules. So what are the requirements to get a PLA code? So for a test, you have to receive a PLA code. It must be commercially available in US. So it must be a real test doctors can order for patient care. So second is the lab or manufacturer must apply for the code. So PLA codes are requested by the company, not owns or performs the test. So where these are category 1 codes then, so if a similar test is widely used, it meets the category 1 standards, then it will be coded in the regular CPT 80K series, not as PLA. So if a PLA code exists for a test, you must use a PLA code. No double coding. The service should not be reported with any other CPT codes. So you cannot build this additional molecular codes for the same test. The PLA code fully describes the service. And this code, it encompasses all analytical services required for the analysis. So what do PLA codes include? So a PLA code is all inclusive for the lab test analytical process. So that means the code already covers steps like cell lysis, nucleic acid stabilization, extraction, digestion, amplification, hybridization, and detection. So you do not separately bill for these internal lab tests. They are bundled into a single PLA code. So what can be billed separately here? When can be billed separately? So if there is a separate distinct procedure done before the actual molecular test, like microdissection, for example, CPT code 88,380, 381. So special specimen preparation before lysis. So those can be billed separately because they are not a part of PLA's built-in analytic workflow. So how and when PLA codes are released? So PLA codes, they are updated more frequently than many other CPT sections. So they are published electronically on the AMA CPT website. So they are released in quarterly CPT data files. At minimum, they also appear annually in the CPT code book. So why this matters? Because new proprietary tests come out fast. So PLA codes keep up. So coders and labs must check the updates regularly, not just the yearly book. And Appendix O, the name list. So Appendix O is like a cross-reference list. It links the PLA code number, the exact proprietary test name. So if you know the brand or test name, you can look it up in the Appendix O to find the correct PLA code. So you must match the exact test first. So you can't use a PLA code unless the lab test performed is exactly the proprietary test. So the method and also purpose match the code descriptor. So PLA codes, they are generally test specific but not close enough codes. So if it's not the exact branded test, you should not use that PLA code. And what if more than one PLA code looks the same? So when more than one PLA has an identical descriptor, the codes will be denoted by the symbol. So sometimes different companies have tests that sounds the same. So CPT book marks this with 2 stars to show the descriptor test may be identical but the proprietary test name is different. So you have to always check the appendix O and the test actual name but not just the descriptor wording. And final note on PLA section. So if a test qualifies as a PLA, it will have a dedicated PLA code and it lives in the PLA section, not in the regular CPT molecular pathology code sections. So any PLA code test satisfies category 1 criteria. So some proprietary tests, they start life as a PLA code. If that test later, they meet the category 1 CPT criteria. This is accepted by the CPT editorial panel. So then the PLA code gets an N symbol added to it. So even with the N, the code stays in the PLA section. So the N is a flag here. So this test now it meets the category 1 standards. So now what if a proprietary test is already accepted for category 1? So a test is approved in principle for a category 1 code, but the actual category 1 code number isn't published yet. So if the lab applies for a PLA code in the meantime, the PLA application takes priority and the test will be coded as a PLA first. So remember, PLA codes are a faster way to get a billable code while waiting for the slower category 1 process. So where does the code live and who maintains it? So PLA codes, they only appear in the PLA section. So the responsibility is the company or lab that owns the test. They must keep the test description accurate and ensure the code still matches what the test actually does. So if the test changes but the code description isn't updated, so coding becomes inaccurate and risky. So when is a new PLA code required? So if you add more targets, for example like DNA, RNA, protein. So if the original PLA test tested say 5 genes and now the company adds more genes or proteins, this is not the same test anymore. So in these cases, you must apply for a new PLA code. And second is if the test name changes because the test changed. So if the test method, performance or the score changes enough that the test name changes or the clinical purpose changes, so then it's considered as a different test and the new PLA code required. So this is how you have to use PLA codes and when they are required. So this is for today's session. I hope this session has helped you. So the next session, you're going to learn CPT coding guidelines for medicine section. Thank you.